Welcome! We're doing a deep dive today on Molluscum Contagiosum Virus, and this one is specifically for our colleagues in medical microbiology. Just so you know, all the information we're covering was compiled using a Gemini deep search, and if you want to get into the details, you can find a full list of sources in the linked Notebook LM document.
Okay, let's get right into this, because this quote really hits on the central paradox of this virus. You've got what is, clinically speaking, a pretty minor bug, right? But for us, it presents these huge microbiological and therapeutic challenges that we're grappling with all the time.
And this really shows you the split personality of Molluscum Contagiosum Virus. From a clinical perspective, especially here in the Netherlands, it's just waterwratjes, waterworts. Benign, self-limiting, annoying but not dangerous. But from our side of the bench in the lab, it's this incredibly tough opponent. So what makes it so difficult?
To really get a handle on those challenges, we've got to profile the culprit itself. We need to understand what makes this particular pathogen so unique.
So let's take a look at its taxonomy, because this is where you start to see just how unique it is. It's a poxvirus, sure, part of the Poxviridae family. But here's the kicker. It is the only species in its entire genus, Molluscum Poxvirus. That sets it way apart evolutionarily from its more famous cousins like variola or monkeypox virus.
And structurally, it's a giant. It's one of the largest viruses that infects humans, with this classic, complex, brick-shaped look. But the real genius here is its strategy. It makes two different infectious forms. You've got the intracellular mature virion, the MV, which is tough and built for spreading between people. Then you have the extracellular enveloped virion, or EV, which is perfect for stealthy cell-to-cell spread inside a single person. It's a brilliant, two-pronged attack for transmission and persistence.
Okay, so all of this unique biology isn't just academic. It has huge, real-world consequences, especially when we try to diagnose and treat it.
This right here is the key. If you take away only one thing today, let it be this. The fact that this virus flat out refuses to grow in the lab is the root cause of almost every single one of our problems with.
And this culture block, well, it doesn't just put the brakes on research. It actually sets a really specific and honestly, a dangerous trap in our diagnostic workflow.
Okay, so here's the trap. You get a clinical sample, you put it on a common fibroblast cell line like MRC5, and you see a cytopathic effect. You see the cells rounding up, ballooning, and you think, aha! But the problem is, what you're seeing looks exactly like Herpes Simplex Virus. It's a major diagnostic pitfall. The only clue is that this effect is abortive. You can't serially passage it.
And, this whole unculturable thing has thrown a massive wrench in antiviral development for decades. And that has led to this huge split in clinical philosophies, which is really obvious when you look at the approach right here in the Netherlands.
And this is where you see that huge split in clinical thinking. The Dutch guideline from the NHG is crystal clear: no treatment recommended. Now compare that to the international view, say from the CDC, where active treatment, things like cantharidin, curettage, cryotherapy, is seen as a perfectly valid option. It's a fundamental disagreement on how to manage this. Now, the Dutch afwachtenbeleid, this wait-and-see policy, it isn't passive. It is a very deliberate clinical choice. And the logic is sound. The infection is benign. It's going to go away on its own. So why put a child through painful treatments that could even cause scarring when the best management is just voorlichting en advies, good old information and advice?
So you might be thinking, how do we get past this roadblock? Well, the virus might not want to cooperate, but science is finding some pretty clever workarounds.
When it comes to diagnostics, the answer is, of course, molecular. Nucleic acid amplification tests are the gold standard now. By targeting conserved genes like P43K, we can get highly sensitive, highly specific detection, and we can even genotype. It completely sidesteps the need for culture, and that's how we avoid that Herpes Simplex Virus mimic trap.
But that still leaves the million-dollar question. What about developing new drugs? This has been the real challenge for years, and this is where a truly elegant, just a fantastic piece of molecular virology comes into play.
I mean, check out how brilliant this is. Researchers took the Vicinia virus, which grows beautifully in culture. They knocked out one of its essential replication genes. Then, in its place, they spliced in the equivalent gene from Molluscum Contagiosum Virus. And what you get is this viable, chimeric virus whose replication now depends entirely on the molluscum protein. It gives us a functional proxy, and that finally opens the door to high-throughput screening for targeted antiviral drugs. It's a total game changer.
All right, so with these incredible new tools in our back pocket, let's bring this all back to the real world, to practical applications and what the future might hold for managing this very persistent virus. You know, when it comes to prevention, it's actually pretty straightforward, and these key strategies line up perfectly with that Dutch focus on advice and practical management. It's all about avoiding direct contact, not sharing things like towels, and really importantly, advising patients to cover the lesions. This helps prevent both spreading it to others and spreading it on themselves. Simple, effective, and no aggressive intervention needed.
And this really brings us to a final, and I think a really interesting question for the future. We are just now entering an era where targeted drug discovery for Molluscum Contagiosum Virus is actually possible. So could these new antivirals, if they prove to be effective and painless, could they be the thing that finally bridges that deep philosophical gap between the Dutch wait-and-see policy and the more active international guidelines?
We'll have to wait and see on that one. Please subscribe for more deep dives like this one. And look, as our colleagues in the field, we would genuinely love to hear your thoughts and insights on this, so please leave your feedback in the comments. Thanks for watching.